Hello, hello. Hello, hello. Good morning. Okay. Okay. Can hear me, yeah? No problem, yeah? Okay. Yeah. So yesterday we already finished chemistry. Okay. Unit 2. So we got started with biology, unit 3. Yeah. Okay. Okay. So we started with diffusion and then osmosis. Okay. And then today we're going to continue with active transport. Yeah. Okay. Yep. Okay. What is active transport? Okay. So yesterday we already learned about diffusion. Okay. So diffusion is going from high concentration to low concentration. Okay. So whether there is membrane or not, it will happen. Okay. If we have a different concentration, diffusion will happen. Okay. Osmosis, okay, is going from high water potential to low water potential. Okay. So this one we just only talking about water and then must be going across cell membrane for for osmosis. Okay. Excuse me. Excuse me. Okay. So active transport. Okay. How about active transport? Okay. What's the difference? Okay. So active transport, we compare directly with diffusion. Okay. Compare directly with diffusion. Active transport is from low concentration to high concentration. Yeah, it is opposite. Okay. Okay, so let's see here, this example here. Okay, so this one is diffusion. Okay, and then this one is outside. Okay, outside of the cell. Okay, and then this one is inside of the cell. Yeah, okay. So this is our cell outside. Okay, and then inside. Okay, so we're just talking about the substance going inside and outside of the cell. Okay, so if we look at diffusion, you see, outside is high concentration, inside is low concentration. Okay, so if outside high concentration, inside low concentration, it just naturally it will go inside. Okay, naturally it will go inside. Okay, this is what we call passive transport. Okay, automatic. It will happen by itself. Okay, so sometimes it depends on what substance. Some substance they can get past the cell membrane directly. Okay, and then we have another type which is facilitated diffusion. So some substance they cannot get past the cell membrane directly. Okay, so we have this channel protein that helps the substance to pass through. Okay, no problem. Still, it's going from high concentration to low concentration. Okay, and then we have the third type, which is active transport. Okay, okay. So what it means by active transport? Okay. Now we can see a key difference here. We can see outside is low concentration. Okay. Inside is high concentration. Okay. Outside is low concentration. The diffusion cannot happen because outside is low concentration. Actually, diffusion will happen in this way. Okay. But we don't want that. Okay. So let's say this is a plant cell. Okay. This is a plant cell. So inside the cell, we have high mineral. Okay. We have high mineral ions. Outside, we have low mineral ions. Outside, we have low mineral ions, but as a plant cell, I still need more. I still want more mineral ions. So in this case, even if the diffusion cannot happen, I will try to bring in by myself. Even if the diffusion cannot happen, I will bring in the substance by myself. But in this case, it's not free. Yeah, so diffusion, it will happen by itself. It is free. But active transport is not free because it does not follow the concentration gradient. So I need to use ATP. ATP means I need to use energy. Okay, so this is the concept of the active transport. Okay, even if outside is low concentration, I still want to bring in this substance, but it cannot happen by itself. So I need to use energy to bring it in. Okay, so that is the concept of active transport. Okay, so active transport is opposite of the diffusion and it requires energy. Okay, so that is the key point. Okay, active transport is the movement of particles through a cell membrane from a region of lower concentration to a region of higher concentration. Okay, so basically it's going against concentration gradient. Diffusion, it follows the concentration gradient, but active transport is going against. Okay, and then it needs energy. Yeah, so these are the two big differences we need to remember about diffusion and active transport. Yeah, okay. The direction of movement. Diffusion is high concentration to low concentration. Active transport, low concentration to high concentration. Okay. Whether energy is needed. Okay. So diffusion is no energy required. We don't have to do anything. It happens by itself. Active transport, we need to provide energy. Okay. Yes. Okay. Some more information. Yeah. Animal and plants, they will use active transport to move some substance because the concentration gradient is not always the right way around, like this plant cell. Okay. Yeah, so even if outside the mineral ions is low, the plant is still need more mineral ions. It will use energy to bring it in against the concentration gradient. So the cell needs to provide energy. Okay, and then the energy is provided through respiration, is produced by the mitochondria. Okay, and then another key point is for this active transport, we need protein carriers. Okay, so this is example of a carrier protein at the cell membrane. Yeah, okay. So this carrier protein is like a gate. Okay, so it can open and close. We can control this gate open and close to bring in the substance that we want. Yeah, okay. So it will move particles across the membrane and then it uses energy to move the particles against their concentration gradient. Yeah, so even if outside concentration is low, it is still able to bring in the particles that we want. Okay, yeah, so some example of active transport. Yeah, example, root hair cell, what I mentioned just now. So outside, we have low concentration of mineral ions. Okay, yeah, okay, so let me just make it bigger. Okay, so outside we have low concentration of mineral ions. Okay, inside we have high concentration. Yeah, normally what will happen is diffusion go outside. Okay, the plants will be losing the mineral ions, but the plants, they want to protect themselves. Okay, they don't want to lose the mineral ions, they want more. So they will use the active transport to bring in the mineral ions that they want, even if outside is low concentration. Okay, yeah, so this is the first case. Root hair cell will bring in the mineral ions such as phosphate from the soil into the roots. Okay, so next one, small intestine. Yeah, small intestine. Okay, so this is our small intestine. If we zoom in, okay, so this is our blood. Okay, this is our intestinal lumen. Yeah, so let's say this is our small intestine. Okay, So we are looking at this place here. Okay. So after we digest everything, we will have lots of glucose in our small intestine after we finish the digestion. Okay. So when this concentration is high, and then inside the blood, the concentration is low for the glucose, the diffusion can just happen. Yeah, the diffusion can happen, no problem. But after some time, we absorb almost everything. Yeah, after some time, we absorb almost everything. Now, inside the lumen is low concentration, and then inside the blood is high concentration. We absorb most of the glucose, but we don't want to waste anything. Even if outside it just left a little bit of glucose, we don't want to um, waste anything. What we will do at this point just now is diffusion when outside is high concentration. But now inside here it is high concentration, outside is low. Now we will use active transport. Yeah, okay. So even if outside left a little bit of glucose, we don't want to waste anything. We will use active transport to bring it in. Okay, so that is when we use active transport for the small intestine. Yeah, small intestine, we will bring in glucose from the small intestine into the blood. Okay, we don't want to waste anything. Okay, next, kidney tubules. So we will learn more about this in later chapters, but just briefly, okay, kidney tubules, how it happened. Okay, so yeah, so this is our kidney. So the function of the kidney is to filter. Yeah, okay. So filter, remove, okay, what we don't want, like urea. Okay, we don't want the urea. And then keep, okay, what we want. Example, glucose. Yeah, okay. So what will happen is, okay, we'll learn more in detail later. First step, it will happen this diffusion, I mean the filtration. Yeah. So it will happen the diffusion. The small molecules will go inside this tube. Yeah, like glucose. Yeah. Glucose, amino acid, mineral salts. Okay. It will go inside here. Yeah. Okay. And then it will go through this tube. Okay. In this process, we need to get back selective reabsorption. Yeah, we need to get back what we want. So actually, we want to get back our glucose. Mostly, we want to get back our glucose, also some amino acid and mineral salt, but mostly we want to get back our glucose. So the glucose will return back to our blood. Yeah, okay. Return back to our blood, and then the urea will just follow this tube, okay, and then it turns to our urine. The urea, we just let it go with our urine, but the glucose, we want to reabsorb back the glucose. We don't want to waste it. Okay, so this glucose reabsorption, we will use active transport. Yeah, because it depends. If the glucose is high concentration, it will come in by diffusion naturally. But if it is low concentration inside here, we can take it back by active transport. So that is the concept here. Okay, how it works in our kidney tubules. So we reabsorb the glucose from the filtrate in the tubule into a blood capillary. Okay. Okay, so next we will look at this sample question. Okay, materials can enter cell by diffusion, osmosis, and active transport. Define the term diffusion. Okay, so this is straightforward. What's diffusion? Net movement of particles from a region of higher concentration to a region of lower concentration due to random movement of the particles. Yeah, okay. So key point from high concentration to low concentration. Yeah, and then at this moment, let me just quickly clarify one point, okay, about diffusion. Okay, so here, okay, we discussed about diffusion at the start. Okay, yeah, so here is high concentration here is low concentration. Of course, we will have movement from here to here. Okay? Yeah, okay. We will have movement from here to here. But at some time, we also have this opposite movement sometimes. But it's just that this one will be more. Okay? This one will be less. So that's what we call net movement. So we always see this word net movement. Okay? So it's telling us that the particles, they are always moving. They are always moving. They never stop. Okay? So even if we have this diffusion from left to right, we have this diffusion, but sometimes we have a little bit the other way around. So let's say this 1, 2, 3, 4, 5, 6, 7 in this direction, and then 2 in this the other direction. Our net movement will be 7 minus 2 equals to 5, something like that. Overall, it's still moving to the right hand side, but occasionally we still have a little bit moving to the left. That's what we call net movement. So we cannot think that it's only left to right. Okay. Of course, we have left to right. It's more left to right, but we still have little bit right to left. Okay. So after we reach this equilibrium, we're not saying that it just completely stopped. Okay. So it does not stop. It's still moving, but it's just that left to right and right to left is the same. Yeah, okay. So we are saying that left to right and right to left is the same. Okay. Yeah. So both direction, same amount of movement. Yeah, okay. So we are not saying that they stop moving. It's just that they move same amounts. Okay. So left to right, 3, right to left, 3, net movement, 0. Okay. So that's the concept about net movement. Okay. So at any time, we have movement from both directions. It's just that for diffusion to happen, we need to have net movement in one direction. Okay. So when it reaches the equilibrium, it does not stop. It's just that in both ways, they move the same amounts. Okay. Coming back to our question. Okay. Okay, so this is what we mean by net movement. Okay, next. Okay, describe how the following process differ from diffusion. How the osmosis different from diffusion. How the active transport differ from diffusion. Okay. Okay, osmosis, we only talking about water. Okay, and then for osmosis to happen, we need a partially permeable membrane. Diffusion, it can happen with both, with or without partially permeable membrane. Okay, active transport, it needs energy. Okay, diffusion, no energy. Yeah, so it will move against concentration gradient. So diffusion, high concentration to low concentration. Active transport the other way around, low concentration to high concentration. Yeah. Okay, now we will be looking at a question here. Okay, a potato was set up shown in figure 3.3. Okay, so this is when we start the experiment. This is when we end the experiment. Okay, it was left for several hours. So this is what happened after several hours. Okay. So what is actually happening here? Okay, so we have a salt solution here, and then here we have water. Yeah, okay. So let's say this salt solution is 5% salt, it will be 95% water. Okay, so this is simply water. So this is 100% water. So we are saying that here, the water potential will be higher. Okay, so higher water potential. And then here it will be lower water potential. Yeah, so this is how we compare the water potential. 95% water, this is 100% water. So we are saying here, this is lower water potential. So what we expect will happen, water will move from lower water potential. I mean, it will move from high water potential to low water potential. So that's why we see here the water level increase. Yeah, okay. So the water level increase because of the difference in water potential. So basically the water will keep moving until both sides same water potential. So this solution will get less and less, okay, concentrated. Yeah, less and less concentrated, close to the concentration of water. Yeah, okay. So this is what we observe. So for this kind of experiment, this is what we observe, the movement of water. Okay. So if we have another situation, this one is water here and then salt solution here, then water will go the other direction. Then the water level will decrease on the other hand. Okay. So this is what we expect for this experiment. Okay. So we can take a look at the question. Yeah. So first question, describe what happened to the water in the dish. So we expect the water in the dish, it will decrease. Okay. So because the water move inside to the potato. So we expect this water in the dish will be decreasing. And then in the hollow of the potato, it will increase. Yeah, okay. So this is what we expect our observation. So we supposed to understand what happened in this experiment and suppose we can describe what happened. Okay. So the volume in the dish decrease. Yeah, so here decrease. Volume of the salt solution in the hollow increase. So here it increase. Okay. Next, name the process that is responsible for the changes. We know that this process is the movement of water, which is osmosis. Yeah, okay. Next, explain why these changes have occurred. Okay, so this is exactly what we discussed just now. Okay, okay, let me just copy this. Okay, and put here. Okay. Yes. What changes have occurred? Okay. Yeah. Yeah. So we need to have these 3 points. There was higher concentration of water in the dish than in the potato, for there was a higher concentration of salt. Okay. Yeah. So we can discuss in terms of water potential or concentration. Yeah. So we are saying that here we have higher water potential. Here is lower water potential. So water will move into the potato, going from a high concentration of water to a low concentration of water. So we can say high water potential to low water potential by osmosis. Okay, so these are the key points that we need to include. Okay, next, where does this process occur in the plant? Okay, so where can this happen in the plant? Okay, so basically it can happen in the root. Okay, so when we have the roots we expose to water, water will move into the roots by osmosis. Okay, so what is the importance to the plant of this process? Why this process is so important for the plants? Okay, so basically it helps the plants, enables the plant to absorb water to maintain cell turgidity or to replace water lost by transpiration. Yeah, so basically it helps the cell to get enough water. Okay, question 2. A cylinder of potato of mass 20 gram was placed in a concentrated sugar solution. When reweighed, the mass had a mass of 18.5 grams. Calculate the percentage change in mass of the potato cylinder. Okay, so what is the situation that we have is this. Okay, so let me yeah. So okay, so this is a potato cylinder. Okay, so this potato cylinder, we put in the concentrated sugar solution. Okay. Yeah. Okay. Let me just make it bigger. Okay. Okay. So let me just make it bigger. Okay. So this is a potato cylinder. Okay. So we put in a concentrated solution. Okay. Concentrated sugar solution. So let's say this is 20% sugar. Okay. So this will be 80% water, for example. Okay, so this example of concentrated sugar solution. So inside the cell, normally inside our cell, we will not have concentrated solution. Inside our cell is maybe like 3 or 5%, something like that. Okay, so let's say this is like 5% sugar. Okay, 95% water, for example. Okay, so now straight away we can compare the water potential. So here that will be high water potential, 95% water. So here it will be low water potential. Yeah, low water potential. What will happen? Okay, what we expect to happen, water will move out of the potato cylinder because inside water potential is higher. Okay, so water will move out of the potato cylinder. So what we expect to happen, potato cylinder, the mass of the potato cylinder will decrease. Okay? Because it's losing water. Yeah, okay. So that's why we can see here it goes from 20 grams to 18.5 grams. Okay, 20 grams to 18.5 grams. Okay, the mass of potato cylinder decrease. That's what we expect to happen. Okay, so first question, calculate the percentage change in mass. Okay, yeah. So this kind of question can come up quite a lot. How do we calculate? Step one. Okay, we find out how much the mass decrease. 18.5 minus 20, it decreased by, okay, 1.5 grams. Okay, and then now we calculate the percentage. Yeah. So at first it is 20 grams, decreased by 1.5 grams, multiply 100%. So we end up with decrease 7.5%. Okay. Yeah. So this is how we calculate. If the mass decrease, our answer is negative. If the mass increase, our answer is positive. Yeah. Okay. So let's consider another situation. So let's say this potato cylinder, same potato cylinder. Okay. And then we put in, okay, let's say we just put in water. Okay. So this is 100% water. Okay. And then this one inside the potato cylinder is still 5% sugar and then 95% water. Okay. So now straight away we can see that outside the water potential is high, inside the water potential is low. Okay. So what will actually happen in this case? Now water will move inside the cell because outside is higher water potential. Okay. Water will move inside the cell. I mean the potato cylinder. So now in this case, we expect the mass of the potato cylinder, it will increase. Okay. So for example, now it might go to 20 grams to 21 grams, for example. Okay. So yeah, it depends which solution we put the potato cylinder into it. It will either lose water or it will gain water. So in this case, it will lose water, okay? Because the osmosis is going from potato cylinder to the solution. Okay, next, explain why the potato cylinder lost mass. Okay, why? What we mentioned just now. Water has left the potato cells by osmosis because water potential was lower outside the cells than in them. Okay, so outside is lower water potential, inside is higher water potential. So water will move out. Okay, next. Okay, so here we have part of a root. Okay, yeah. So this is the structure of a root, straightforward. Explain how the presence of the root hair cells on the root enables the efficient absorption of water and mineral ions. Okay, why these root hair cells, they are very useful, very effective. Okay, yeah. So we can take a look at the explanation. Okay. So we can say that we have large numbers. We have many root hair cells, okay, at these plants. And then these cells have large surface area. Yeah, okay, because they have this extension. Yeah, this protrusion. Okay, they have large surface area. And then they have mitochondria. Yeah, so they have this mitochondria to provide energy. Okay, provide energy for this absorption of mineral ions because sometimes they need the active transport to absorb these mineral ions. Okay. Next, root hair cells absorb mineral ions by active transport. We know that. Define the term active transport. Okay, so what it means. So we need to remember the key points about active transport. Okay, so absorptions of particles into a cell across a membrane. Okay, so we also need to have cell membrane for active transport to happen. And then against concentration gradient. Yeah, it's opposite compared to diffusion. And then it is a process that using energy. Okay. Part 2. Explain why respiration rate may increase in the root hair cells during uptake of mineral ions. Okay, so when we are having the uptake of mineral ions, we are using active transport, we need more energy. Yeah, okay, so we need more energy, so we will be increasing the respiration rate. Yeah, okay, increasing the respiration rate that requires energy. Okay, next. Okay, question 4. Complete the table by stating the process involved in the movement of material in each description. Yeah, okay. Okay, so this one should be straightforward. Uptake of water in plant by root hair cells. Okay, we know uptake of water straight away. We are talking about osmosis. Yeah, okay. So we are talking about osmosis. Okay, next. Loss of water vapor from the stomata of a leaf. Okay, so what is this one? Okay, let me copy the picture. Okay. Okay, so we are talking about this situation. Okay. Okay, so this one, loss of so we can be losing oxygen, we can also be losing water from the stomata of the leaf. Okay, so this one we will be careful. So this one is movement of water, but this is not osmosis. No, this is not osmosis. Why? Because it's not going through cell membrane. Osmosis, it must be going through cell membrane, then it is osmosis. Okay, so if just going through the stoma, with that there is no cell membrane. The stoma is just an open hole. Okay, It's just an open hole. Okay, no cell membrane. Even if it is movement of water, if no cell membrane, we still say that it is diffusion. Yeah, okay. So this part we need to be very careful. Even if movement of water, if there is no cell membrane, it is still diffusion. Yeah, okay. Okay, coming back to our question here. Okay, yeah. So this one, it will be diffusion. Okay, next. Uptake of mineral ions by protein carriers. Okay, so as soon as we see these protein carriers, we know that we need energy. Okay, so if we need energy, means this is active transport. Yeah, okay. Okay, so with this, we finish all the notes for the unit 3 of the biology. Yeah, okay. So we finish all of unit 3. Okay, yes. Okay, so biology unit 3 already finished. So now we will move on to the workbook. Yeah, okay, so we'll be going to the workbook. Okay, let's take a look. Yeah, so we'll go through the workbook questions. Should be very straightforward. Okay, so first one, define the term diffusion. Okay, so what it means by diffusion, give 3 key points. Okay, we remember that. Okay, yes. So net movement of particles, region of from region of higher concentration to a region of lower concentration. Yeah, so basically it's going down a concentration gradient as a result of the random movements of the particles. Okay, so we can see here no mention about cell membrane. Okay, so it does not require a cell membrane. So whether cell membrane is there or not, diffusion can happen. Okay, next. In the lungs, a substance diffuse from alveoli into the blood capillaries. Okay, so we can come back and take a look at the notes. Yeah, okay. Yes. So this is our situation here. Okay, so we can see the direction of movement. Carbon dioxide is from blood to alveoli, oxygen from alveoli to blood. Okay, so here, okay, what the question is asking about, okay, from alveoli into the blood. Okay, so it's this direction here. Okay, so we are talking about movement of oxygen. Okay, state 3 factors that will encourage a fast rate of diffusion for this substance. Okay, so we can talk about, okay, what are the adaptations or features of the lungs, okay, that will make this diffusion of oxygen faster, or will make this diffusion of carbon dioxide faster or more effective. Okay, we already learned that, okay, is this. Yeah, so we did mention, yes. So distance, okay, very short distance because we have very thin moist walls, okay, just 1 cell thick, very thin. Concentration gradient, so there is a difference of concentration of oxygen, okay, so here oxygen is high inside the blood, oxygen is low, so it will diffuse into the blood. Surface area, so we have hundreds and thousands of these alveoli. Okay, so it provides a high surface area. Okay, so in this case, temperature is not too important. Okay, basically we just maintain our body temperature, so that will do. Okay, yeah, so we can answer here the 3 factors. Okay, short distance, high concentration gradient, large surface area, and higher temperature. Okay, so if we have a slightly higher body temperature, then the diffusion will be faster as well. Okay, next. Okay, suggest and explain what would happen to the rate of diffusion if the temperature of the lungs went down. Okay, so if temperature decrease, so the particles or the oxygen molecules, they will have lower energy. Yeah, so they will be having lower energy. They will move slower. Okay, so basically they will slow down. So the diffusion will also slow down. Yeah, okay. So that is the main point. Okay. So rate of diffusion will drop. Okay, diffusion will slow down because molecules have a lower kinetic energy. Yeah, the molecules moving more slowly. Okay, a person's breathing rate increase. Okay, so this is straightforward. We breathe faster. Okay, so if we breathe faster, we will be having more oxygen. Yes, yeah. Okay, so if a person breathe faster, so here we will be having more and more oxygen. So the diffusion will happen faster in this case. Okay, because we have a higher concentration gradient. Okay, next. So we expect rate of diffusion would increase because concentration gradient would increase or maintain. Okay, next. Define the term active transport, making 3 key points. Okay, so what is active transport? Straightforward. We know that. Okay, so again, movement of particles through a cell membrane. Okay, so active transport also need a cell membrane to happen. Okay, but this is from lower concentration to a region of higher concentration. So basically, it's going against concentration gradient using energy from respiration. Okay, so this is a key point. Okay, next, we will look at the question. Okay, so let me just copy this. Okay, put here. Okay. Okay, so what is happening here? Okay, we can read the question first. Okay, so we have two plants, A and B, were grown in the soil with different nitrate ion concentrations. Okay, the concentration of nitrate ions was measured in both the soil and inside the roots of the plants. Both plants were found to be successfully absorbing the nitrate. The table show the result. Okay. Okay, so okay, let me just concentration of nitrate ions in the soil. Okay. Okay, so let us just take a look. Okay, so this one is the root hair cell. Okay, so this one is the soil. Okay, here again, this is the root hair cell. Okay, for the plant B. Okay, and then the soil. Okay, so in the soil is 157 here. So here this is 82. So this is high, this is low. Okay, and then here inside the root is 156. Okay, so 156, and then inside the root here is 114. Okay, so yes. So now we need to look at plant A, what process will happen, and then why. Okay, so yeah, you can also think about it, what you think, what will happen. Okay, so we can see here, if we compare inside and outside, outside is 157, inside is 156. So inside is lower. So what we expect, we will have diffusion. Okay, diffusion of the mineral ions going inside because outside is higher. Yeah, okay. So this is what we expect will happen for plant A. Okay, diffusion only because the roots have absorbed nitrate until there is no concentration gradient. Okay, straightforward. Okay, how about B? Okay, so for plant B, okay, inside is higher. Inside is high. Okay. So now what will happen? We will have diffusion outside. Okay. Because inside is higher. So we will have diffusion. Okay. But we will also have something else. We will have active transport going in. We will have active transport going in. Why? Because the plant, it needs to protect itself. If we just let the diffusion happen by itself, in the end, the plant will lose the ions. Okay? The plants will be losing a lot of ions. It's not good for the plant. So the plant, it will do something to protect itself. It will do this active transport to bring in the ions return back, okay, even faster than the diffusion. Okay, so that is the concept for this question. So here, what we expect will happen will be both diffusion and active transport. Okay, so the root have absorbed nitric ion against a concentration gradient, building up a higher concentration than that present in the soil. Okay, yeah. So it needs to work faster than this diffusion, so it does not lose the ions. Okay. So now we have another experiment. The roots of plant B were treated with cyanide, which is a respiratory poison. Explain why the movement of nitrate stopped in the plants. Okay. So this is straightforward. Treated with cyanide, a respiratory poison. Okay. So basically, this cyanide will kill the mitochondria. Yeah, okay. So it will kill the mitochondria in the cell, okay? Or it straight away kill the cell. Okay, respiratory poison. So there is no energy. Okay. So no energy, the active transport cannot happen. Yeah, active transport cannot happen. So the movement of the nitrate will stop. Okay. So that's the key point. So basically, the plants need energy from respiration to carry out active transport. So the cyanide is a respiratory poison. It stops the energy production. So there will be no energy for active transport. Okay. Next. Okay. Which process is responsible for the movement of the substance in the table below into or out of plant cells? Okay. Please tick or cross in each box to show your answer. Okay. So okay, this one, maybe you can try. Okay. So maybe you can try. What do you think? For the oxygen will be diffusion, osmosis, or active transport? Have a try. Okay, based on what you remember, what we learned so far, have a try. So in a plant cell, for the movement of oxygen, it will be happen by diffusion, osmosis, or active transport.
Um, Osmosis.
Osmosis is for water. Yeah,
Oh,
osmosis is for water.
diffusion.
Yes, 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 yes. Okay. So oxygen will be diffusion. Yeah, okay. Okay. Just diffusion. Okay. Not osmosis, not active transport. Okay. Yeah. So as we mentioned just now, water will be osmosis. Yeah. Movement of water will be osmosis. Okay. but sometimes it can happen diffusion, okay, like what I mentioned just now, here, okay, in the plants. If it's through the stoma, the diffusion of water can happen. If going through the cell membrane, it will be osmosis. Okay. Okay. How about phosphate ions? Okay. What do you think about phosphate ions? Diffusion, osmosis, or active transport?
Um, Active transport.
Correct. Excellent. Excellent. Okay. So active transport and also sometimes diffusion. Okay. As we saw just now, if you see inside concentration is lower, then diffusion can happen. Yeah, okay. If outside is higher, then it will be active transport. Okay, carbon dioxide. Okay, what do you think about carbon dioxide?
Diffusion.
Yes, excellent. Okay, so what we mentioned here, carbon dioxide goes in, okay, through the stomata and then diffuse inside the cell also by diffusion. Okay, yes, excellent. Okay, next. Okay, so this is experiment about potato cores. Okay. Yeah. Is um is this experiment here. Yeah, so we cut the potato and then we put in the solution of different concentration. Yeah, okay. So it is this experiment here. Okay. Yes. So what is going on here is okay. Um Okay, six identical potato cores, so means they are the same. They are cut, so they have the same mass and same surface area. Yeah, like this one. Okay, each was placed in the sugar solution, okay, of different concentration. So we can see here 0, 0.0, 0.2, 0.4, 0.6, 0.8, 1.0, okay? Increasing concentration and left for 6 hours. Okay. So if we did this, we know osmosis will happen. Yeah, osmosis will happen. And then, so either the potato, it become lighter if it lose water. Okay. And then it become heavier if it gain water. Okay. So it depends on the osmosis. Okay, so we will reweigh and the percentage change of the mass was calculated. The graph shown the result. Okay, yeah, we can take a look. Okay, at the graph. What is happening? Okay, so they're asking zone A, zone B, and zone C. Yeah, zone A, zone B, and zone C. Okay, so what is happening? Okay, so okay, so zone A, the mass increase. Okay, so what's happening here? Okay, this happened when we put in low concentration. Yeah, when we put in low concentration of solution, like here, 0, 0.0, 0, 0.2. When we put in low concentration, okay, what is happening inside the cell? Okay, so when we put low concentration, okay, so let's say this is like 1%, okay, sugar, example, 99% of water, okay. So inside here, it may be like 3%. 3% sugar, okay, 97% water. Okay, so we can see here outside is 99%. So we will be having higher water potential outside, inside is lower water potential. So water will move from high water potential, okay, water will move from high water potential to low water potential. Yeah, so water will move inside, okay, so the mass will increase. So that's why we see here the mass is increasing, okay. So let's say we start with 20 grams, so maybe it increased to 23 grams, something like that, okay. So the mass will increase, okay. How about zone B? Okay, so what's happening in zone B? Okay, so it's the opposite situation. Okay, so zone B. So inside the cell is still 3% sugar and then 97% water. And then outside is high concentration. High concentration. So example, we have 10% sugar, okay, 90% water. So we can see here, inside is high water potential, outside is low water potential. So we expect the movement of water will be going out, okay? Going out of the cell, water going out of the cell. So we expect the mass will be decreasing. So that's why we see the mass decreasing, okay? And then we see here, it become flat horizontal line, okay? So flat horizontal line, it means it lost all water that it has. It cannot lose anymore. Yeah, okay? Okay, how about C? Okay, so what is the situation for C is, okay, so this is what we call isotonic, okay? Isotonic solution means inside and outside same, okay? So let's say outside is 3% sugar, okay, 97% water. Inside also 3% sugar, okay, 97% water. So inside and outside same. The water potential is same, okay? So means there will be no net movement, okay? So that's why no net movement. The size of the potato cylinder, the mass, it remain the same, okay? So the mass, it remain the same. So that's what happening in this graph, okay? How we understand what is happening in this graph, okay? So if we look at the explanation, zone A, yeah, okay? So sugar solution is becoming less concentrated, yeah, okay? Lower concentration. The water will enter the potato cells by osmosis, yeah, so we see here, water enter. So the tissue will gain mass, mass will increase. The more dilute the sugar solution, the more mass will be gained. So you see, going from 0.2 to 0, the mass increase more. Okay. Okay, zone B is the opposite, what we mentioned just now. Okay, sugar solution become more concentrated. Yeah, so we can see here, it's becoming more concentrated. Okay, no water leave the potato cells by osmosis, so the tissue loses mass. Okay, the more concentration treated the sugar solution, the more mass is lost. Okay, so it's losing water. Yeah, going out. Between 0.6 and 1.0, there is no additional mass loss as the potato cells have been fully flaccid in each case. Okay, so we can see here 0.6 to 1.0 is almost flat, the graph, because basically it lost all the water that is available. So Even if we put in more concentrated solution, it does not lose more water. Okay. Now, zone C, which is this point, where there is 0% of mass loss. The mass, it just remain the same. Okay. So what's happening here? Okay. There is no net gain or loss of mass by potato because the water potential inside and outside, it is the same water potential. Okay. So there is no net gain or loss of water. So osmosis does not really happen in this case. Okay. Yeah. So that's how we understand it. Okay. Next. Which set of statement correctly describe active transport? Okay. So what we mean by active transport is this situation. Okay. High concentration to low concentration. No. Low concentration to high concentration? Yes. Energy required? Yes. Energy not required? No. Okay. So that is the concept of the active transport. Okay. Next. Okay. Cell can get substances by diffusion, osmosis, or active transport. Complete the table about absorption of substance in two types of cell. Okay. So root hair cell, okay, if it wants to get the water, how? Okay, so it will get by osmosis. Yeah, okay. So it will be going from high water potential to low water potential. Energy is not needed. Okay. How about for nitrate? Okay. So when the plants want to get nitrate, what are the ways to get it? Okay. So it can get by diffusion or active transport. Okay. So like what we mentioned just now. Okay. So when inside is high, outside is low, then we will have we will need active transport, okay, to bring it in. Okay. How about when outside is high, inside is low, then we will have diffusion. Yeah, okay. Diffusion is from high to low. No energy is needed. For active transport, outside is low, but we still want to bring it in, then we will be using the active transport and using energy. Okay. Wheeler cell in the small intestine. Okay. As what we mentioned just now, okay, in the notes. Eh, yes, this situation. Okay. Okay, Wheelers in the cell. Okay. So how it happened? Okay. Okay, it is two situation as what we mentioned just now. So first, if just after, okay, just after digestion, okay, we will have lots of glucose. Okay, we will have lots of glucose in the intestine, but low glucose in the blood. Okay, so in this situation, it will just happen by diffusion. Okay, automatically. Okay, so it will happen by itself. Okay, so just like this, diffusion from high concentration to low concentration, no energy is needed. Okay, how about after some time, we already absorb most of the glucose. Now the glucose is low, and then in the blood, it is higher glucose. But we don't want to waste any glucose. We just want to take everything. It is food, it is energy. We don't want to lose it. So we will take it by active transport. Yeah, we will take it by active transport. So both situation can happen. Okay, active transport, low to high, and we need energy for that. Okay, next. Okay, yeah, so this one is another experiment of the potato cores. Okay, 6 potato cores were all cut to the same length and then placed in a range of sugar solution. After 1 hour, the cores were removed and remeasured. The results are shown in the table. Yeah, so sometimes we check whether the mass decreased or not, 20 grams to 18 grams. Okay, we can also check the length. Okay, so example, if it lose water by osmosis, it will become smaller. If it lose water, if it gain water, it will become bigger. Yeah, okay. So it's the same experiment. Yeah, this one. It's still this experiment, same. Yes. Okay. So for this question, I would like you to practice calculation, the change in length and the change in percentage. Okay. So you can try to finish the calculation and then we will discuss when you are ready. Okay. So you can remember how to do the calculation. Yeah. So if we get shorter, okay, our change will be negative. If we get longer, it will be just positive. Okay. The percentage also. If it gets shorter, it will be negative. The longer it will be positive. The percentage change. Okay. So you can try to do the calculation. Yeah. Once you are ready, we can check the answers. Okay. Yeah. Okay. Maybe I just demo. Okay. Demo for one. Okay. Okay. Change in length. Okay. So we're going from 5.0 to 5.3. So essentially, we increase 0.3. So we will be saying plus 0.3 here, increase in length. Okay. How about percentage change? Okay. So we change by 0.3. Our initial length is 5 compared to our initial length multiply 100%. Okay. So that will give us, okay, so we calculate that 0.3 divided by 5. Okay, 0.3 divided by 5 multiply 100%. Okay, so we are saying that this one is plus 6%. Okay, makes sense, yeah? Can understand, yeah? Okay, now you try to calculate the rest. Okay, you try to calculate for this 0.2, 0.4, 0.6, 0.8, 1.0. Okay, so you try to do the calculation. Okay, have a try.
0.2 is plus 0.1.
Mm-hmm.
And the
And
percentage is 2%.
correct, excellent. Okay,
Minus 0.3.
yes.
So minus 6.
Okay, excellent.
Minus 0.6.
Mm-hmm.
So minus 12.
Yes. Okay.
0.8 and 1.0 are the same. Minus 0.7 and minus 14.
Okay, excellent. Perfect, perfect. Okay. So next part, okay, so we already done A and B, percentage change in length, percentage change in length. So now we will be plotting a line, okay? So we'll be plotting a line, okay? Um, okay, so we need to see carefully what they want us to plot. Percentage change in length and concentration. Yeah, okay. So concentration in the x-axis, okay? So which means we want to plot this one, okay, in the x-axis, okay? So which means we will plot this in the y-axis. Yeah, so step one, we just need to make sure we are plotting at the correct axis. Okay, so step 1, we will okay, so they already give us some guideline where to put the axis, positive and negative. So means we put here the axis. Yes, okay. And then here the y-axis. Okay. And then we label. Okay. So the x-axis will be this sugar concentration mole per dm cube. Yeah. Okay, so we can put somewhere here. Okay. And then the y-axis. Okay. So if you already very clear, I can skip it also. Yeah, but I will go through the steps. Okay. Okay, now okay, let me just copy the table. Okay. So 0.2 from 0 to 1.0. Yeah. So if you think it's like too easy, you want me to skip, you just let me know. Okay. Otherwise, I just go through the step by step. Okay. And then okay. Yeah, so we just put side by side. Okay, so help us with the plots. Okay, as we go through the step. Okay, so now we want to set up the scales. Okay, so make sure we do the scale properly. Okay, so I think this one is straightforward. For the concentration is just 0 here. Okay, 0 0.2, 0 0.4, 0 0.6, 0 0.8, and then 1.0. Okay, easy. Okay, percentage change in length. Okay, so either we can do 2, 4 here. Okay, but we cannot do that because um this is 6. So we need to do 5, 10. Yeah, okay, so we'll do 5, 10. So here will be um -5, okay, -10, and -15. Okay, so it's done. So 0 0.0 is 6, okay, and then 0 0.2 is 2, okay, 0 0.4 is -6, okay, which is somewhere here. Okay, 0 0.6 is -12, somewhere here. Okay, 0 0.8 is -14, okay, 1.0 is -14, okay. So, okay, yes. So we can draw a smooth curve, something like this. Try to sketch a smooth curve that connect all of the points. Okay, so as smooth as possible. Okay, so this is what we expect to draw. Okay, and then yeah. So if we look at the marking scheme, yes. Okay, so if we done everything correctly, so it will be something like this. Okay, that's good. Okay, next, use the graph to predict which sugar solution there will be no change in length. Okay, yeah. So just now we already discussed how to get that is here. Yeah, when there is 0% of the change in length. Okay, so if we check carefully, so this is 0.2. Okay, so we have 5 gaps. So basically we have 5 gaps. It is equal to 0.2. Yeah, so 0.2. So 1 gap divided by 5 divided by 5. So 1 gap will be equal to 0.2 divided by 5. Okay, so it will be 0.04. Okay, so we have from here to here is around 1.5 gap. Okay, so 1.5 gap, 1.5 times 0.04. Okay, that is 0.06. Okay, so which is telling us that this point here is 0.2 plus 0.06. Okay, so which give us 0.26 mole per dm cube. So which means the concentration where there is no change in length, it is at this concentration here. Okay, so it's telling us that at this concentration, the water potential inside and outside the cell is the same. Okay, so that's how we analyze this graph. Okay, so our answer is 0.26. Okay, yeah, so we get that. Next, explain why there will be no change in length at this concentration. Yeah, we just explained that just now. Why at here, no change? Okay, because concentration of sugar in the potato cell is same in the surrounding solution and inside the cell, inside and outside same. Okay, so there is no net movement of water molecules. Okay, so as I mentioned just now, this no net movement is telling us there is still movement. It just inside and outside is the same amount. Okay, so that means there's no net movement. Okay, name the two variables that should have been controlled in this investigation. Okay, so we are very familiar with this. Whenever we are doing experiment, we only can change one thing at a time. In this case, we only changing concentration. All the rest of the parameters must be the same. Okay, if we change more than one thing at a time, if we get a different result, we don't know what caused that change. Okay, so we are only changing one thing at a time. So in this case, we are only changing the sugar concentration. Yeah, so what are the things we must keep the same? For example, the starting length must be the same. We must use the same size of the potato, and then the time, 1 hour. All of them must be 1 hour. We cannot sum 1 hour, 2 hours, then the result will be inaccurate. Okay, so this is what we call control. Control means other than what we change, we only change one thing. All the other things must be constant. Must be constant. Okay, yeah. So leave all costs in the same amount of time, 1 hour. Use the same volume of solution in each test tube. Yeah, like just now. No. Just now we saw the test tube. It must be the same. Yeah. Okay, hold on. Wait. Yes. Okay. We must put all potato cores in the, all the potato cores same size. At the beginning, they put in the same amount of solution. Yeah, so all of this must be constant. Okay. Cut all the cores to the same diameter. Yeah, must be the same size. Okay, suggest one way of making the result more reliable. Okay, so it always can happen. Okay, the the question asking us how to improve the experiment. Okay, if possible, we always try to improve the experiment. Okay, okay, what can we do? Okay, set up 2 or more cores for each concentration. You see, this now, this one concentration, we only do the experiment once, okay? So if we do it 2 or 3 times, we will get more accurate result. Yeah, okay. Measure the length of the course in mm. Yeah, okay. So just now we measure in cm, okay? So if we measure in mm, it can be more accurate. Okay. Weight the course instead of measuring length. Okay. So if we weigh, so let's say we measure the weight, we can measure like 20.2325. Okay. So if we can measure in more with the 4 decimal place, then it can be more accurate than measure in mm or cm, for example. Okay. All right. So now we are done the workbook. Okay, for unit 3 as well. Yeah, okay. So the workbook is also done. Okay, so now we are making the progress here. Okay, so this is also done. Okay, yes. Okay, this is good. Now, yes. So today, biology, we already finished all for this unit 3. So now we will move on to chemistry. Okay, chemistry unit 3. Okay, so this is our plan here. Okay, so means this in progress here. Okay, so now we are moving to chemistry unit 3. Okay, hold on. Yes. Okay, chemistry unit 3. So again, we will start with the textbook. Okay. Okay, so maybe we don't have enough time to finish all, but we will get started. Okay, so this is our progress. Okay, chemistry unit 3. Okay, close all. Okay, let me open the chemistry unit 3. Yeah. Okay. Yes. Okay, so feel free to stop me to ask any question. Okay, or if you need a break or anything like that. Okay, just stop me. Yes. So this is chemistry unit 3. So this is quite a big topic. Yeah, okay. So maybe we not have enough time to finish all. It's a big topic. So basically, this is talking about the different type of bondings, okay, that can happen between atoms, okay? So we'll be learning about different type of bonding. Okay. Yes, this is quite a big topic. Okay, so maybe we can finish tomorrow. Okay, we'll see. So today we will get started. Okay. Yeah. So okay. So the few type of bondings we will look at: covalent bonding, ionic bonding. Okay. Um yes. And then there are two more: metallic bonding. Okay. And okay, giant covalent. Okay. Giant covalent structure. Yeah. So we'll look into total 4 type of bonding. Okay. Yes. So first we will look into ionic bonding. Okay. So what it means by ionic bonding. Okay. So ionic bonding is the attraction. Okay. Attraction between positive and negative ions. Okay. So we'll see here. This is example. Okay. So example, we have a sodium ion, okay, and the chloride ion. This is positive charge. This is negative charge. They will attract. Yeah, okay. So when we have positive ion and negative ion, they will attract. Okay. But how? How we get these ions? Okay. How we get these ions? So this is the big questions. Okay. So once we have the ions, they will attract. Okay. But how do we get these ions? Okay. So this is what we will discuss now. Okay. So for the ionic bonding, we always have a metal plus a non-metal. So always we will have a metal combined with a non-metal always for ionic bonding. Okay. Yes. And then Metal ions. Okay. What will happen with the metal ions? And then what will happen with the non-metal ions? Okay. And then for bonding, okay, any type of bonding or chemical reaction that happens, okay, any type of bonding or chemical reaction only have one objective. Why chemical reaction happen? Okay, we only have one objective. Okay, let me go to the periodic table just briefly. I will show the reason why. Okay, we will take a look at the periodic table. Okay. Yes. Okay. So in the past exam papers, we will be provided with the um, periodic table. Okay. So if we look at the periodic table, okay, um, we have metals and non-metals. Okay. So first, let me just quickly check periodic table. Okay. Hold on. Okay. Yes. So basically, we will know about the category of metals and non-metals. Okay. So the metals and non-metals is the boundary is here. Okay. So this is the boundary. Okay. So here will be metals. And then here, after this line, it will be non-metals. Okay. So all the chemical reaction that happen, they got one objective. Okay. Is to get a full shell. Okay. Whatever they do, what they're trying to do is just to get a full shell. Okay. So we can look at this example here. Okay. This sodium. Yeah. Okay. So we can see this sodium, the electron arrangement is 281. Okay. So total it have 11 proton, 11 electrons. Okay. So the electron arrangement will be 281. So we can see it got one extra electrons. Okay. So what it will try to do, it will try to lose this electron. Okay. So once it lost the electron, it will become 2, 8. It get the full shell. Yeah. Okay. So that's the objective. Okay. Because when they get the full shell, it will become more stable. Yeah. So the full shell is the stable one. Okay. We can look at the example here. If we look at this group 8. Okay. This group 8, they are all full shell. So we can see here, this helium is 2 electrons. Okay, full shell. This neon is, okay, 2, 8. Okay, full shell. Okay, 2, 2, 8. And then this argon, okay, will be 2, 8, 8. And so on. Okay, they are all full shell. So this is what we call noble gas. Okay, noble gas means they are non-reactive. Okay, so they don't like to react with anything. They just stay as they are. They don't react with anything. Why? Because they already have full shell. All of them have a full shell, but all of these they they are not full shell. Yeah. So example, sodium 281, magnesium 282. Okay, and then fluorine 27. Okay, oxygen 26. They are all not full shell. So when they are not full shell, they will try to do something to get a full shell. Example, here they will lose electron to get a full shell. Lose one, lose two, they get full shell. This one, they try to gain electron. Okay. Example, oxygen can gain 2 to get 2, 8. fluorine can gain 1 to get 2,8. Okay, so this one already full shell, they will do nothing. Okay, they just stay as they are. The rest, they don't have a full shell. Either they gain electron or they lose electron to get the full shell. Okay, so that's how we get these ions. Yeah, okay. So this sodium, it will try to lose 1 electron to get the full shell of 2.8. And then this chlorine is 287. Yeah, 287. It will try to gain 1 electron to get the full shell of 288. Yeah, okay. So you can see after each chemical reaction, they always end up with a full shell. Yeah, okay. And then this sodium, it will only lose 1. It will not lose 2, 3, 4. It will not. It will only lose 1. Okay. Because losing 1 will give the full shell. Chlorine is the same. It will only take 1 electron. It will not take 2, take 3, take 4. No, it will only take 1. It just reached the full shell and then it's done. Okay. Yeah, so this is what they will do. So that's why sodium, the charge is always +1. Always. Okay. It only lose 1 electron. Chlorine, the charge always -1. It will not be chlorine -2, -3. No. Okay. Always -1. Okay. Yeah. So, and then to get a balance, okay, to balance the charge, because the ionic compounds, the charge is always balanced. Okay. So 1 positive, 1 negative. So that's why sodium chloride is always 1 sodium, 1 chloride. It will not be 1 sodium, 2 chloride. No. Okay. Will not happen because they always need to balance the charge. Positive 1, negative 1, balance. Okay. So later we will see other examples. Okay, yeah, so this is what's going on here. So the metal atoms, they will always lose electrons to get a full outer shell. Okay, so for example, magnesium is 282. 282, it will lose 2 to get the full shell. So it will form magnesium 2 plus. Okay, another example, aluminium 283, it will lose 3 to get the 28. So aluminium will become 3 plus. Yeah, always. So group 1 here is always +1, the charge. The group 2 is always +2, the charge. Okay. Group 3 is +3. Okay. So we can predict. And then here, group 6, oxygen, sulfur, selenium, and so on. This one, they always receive 2 electrons. They always have 6 outer electrons. They always receive 2. So this one is always -2, the charge. And then this group 7, they always 7 outer electrons. They always receive 1 electron. They always -1, the charge. Okay. So with this knowledge, we can predict. Okay. Let's say I ask you potassium, what charge it will form? Always +1. Let's say I ask you calcium, always +2. Calcium is always +2. Okay, let's say I ask you sulfur, always -2. Let's say I ask you chlorine, bromine, iodine, fluorine, always -1. Okay, so if you know their position in the periodic table, you can predict how many electrons they will lose, and then what kind of charge they will form as a ion. Okay. Okay, now going back to our notes. Yeah, okay. So metal, they will always lose electron. Okay, it could be 1, 2, or 3, depends on the position to form the full outer shell, and then they will form positive ions like sodium +1. Okay. So loss of electrons, we know losing electron is also means oxidation. Okay. We will get into this later. Non-metal atoms, okay, like chlorine, they will gain electron. Yeah, 287, get 1 more electron, it becomes 288. Okay. And then again, it will get the full outer shell. They will form negative ions and ions. when they gain electrons, they become more negative. That's what we call reduction. Okay, so we have reduction here. Okay, example, sodium chloride. Okay, so just now we mentioned. Okay, so sodium have 11 electrons. So the electron configuration is 281. So it will lose one electron to get the full shell. Okay, 2.8, 2.8. And then this chlorine is 17 proton, 17 electrons. So means the electron arrangement is 287. Okay, so it will tend to receive one electron. Yeah, receive one electron to get the 288. So in the end, both of them get the full shell. Yeah, both of them get the full shell. And then this sodium, after losing one electron, it will form positive ion. Okay, so this is how we present it. We must draw this square bracket to present this ion. Okay, so we can draw like this. Okay, or we can draw like this. Okay, so sometimes in the exam, they just want us to put the outer shell. So we just put like this. Okay, in the exam situation. And then sometimes, okay, so for the chlorine, okay, so it will be like this. 1, 2, 3, 4, 5, 6, 7. The 8 electron is coming from the sodium. Okay. Uh yes. Okay. So sometimes in the exam they just ask for the outer shell. We will just draw the outer shell. Okay. Yes. So the chlorine getting one extra electrons from the sodium. So now the charge is -1. Okay. Again, we need to put this square bracket to show it. Yeah. Okay. And then the ion, sodium plus, chlorine -1. Okay. Both of them have a full shell of electron arrangement. Okay. So the sodium will be overall positive +1 charge, chlorine overall -1 charge. Okay. Yep. Okay. And then the ratio, what we mentioned just now, the ratio is always 1 sodium, 1 chlorine. Okay. Why? Because sodium is +1. Okay. Chlorine is -1. So they balance out. Okay. So let's say we have calcium 2 plus, and then we have chlorine. Then in this case, we need 2 negative to balance it. Okay. So we will have calcium and then chlorine, 2 of them. So for calcium chloride, this will be the correct formula. Okay. So, but sodium, it cannot Cl2. No, it must balance the charge. Okay. Yeah. So if we have group 1 and group 7, the ratio is always 1 to 1. Okay. Another example, magnesium fluoride. Yeah. Okay. So if we look at magnesium fluoride, so if we go to periodic table, magnesium is group 2. Yeah. Group 2, it is 282. Yeah, 282. So it will lose 2 electrons. Okay. Lose 2 electrons to get the 28. And then fluorine is 7. Yeah. 27. So it will gain 1 electron. Yeah. So each of them will gain 1 electron. So in the end, we end up magnesium losing 2 electrons, magnesium 2 plus here. Okay, fluorine, each of them gain 1 electron. So full shell, full shell. Okay, so all of them will get the full shell, and then fluorine will become -1, the charge. Okay, so Mg 2 plus, chlorine negative. So means we need 2 of them to balance the 2 plus charge. Okay, so the magnesium will be magnesium fluoride, 2 fluorine. Okay, so this will be the correct formula for magnesium chloride. Okay, yeah. So for the IGCSE, we need to know how to work out the formula of ionic compounds. Okay, so this table, okay, if can remember, it will be helpful. So for the common ions, if we remember the charge, we can work out the formula. Okay, so example, okay, let's say we have potassium, K plus. Okay, let's say we want sulfide, Okay, sulfide S2 negative. So means we must have 2 potassium to balance out the sulfide. So it will be K2S. Correct formula. Okay. Another example, barium hydroxide. Okay, barium is 2 plus hydroxide, negative 1. So we need 2 of them. So it will be barium OH2. Example. Okay. Phosphate. Okay, let's say we have iron phosphate. Okay. So iron is 2 plus, phosphate is 3 negative. Okay, how we balance it? So which means this one we need 3 of them. Okay, so here will be plus 6. This one we need 2 of them. So it will be negative 6. So our correct formula Fe3PO4 2. Okay, like this. So this is how we work out. So if we know the charge, we can work out the formula. Okay, another example, aluminium, oxygen. Okay, aluminium 3 plus, okay, oxygen 2 negative. So which means we need aluminium 3 of them, plus 6, oxygen we need 3. Yeah, negative 6. So the correct formula, Al2O3. Yeah, okay. So that's how we work it out. Okay, so this one, try to memorize if possible, so it can be useful, okay, in the exam situation. Okay, next. Yeah. So this will be some example how to do it, like what we did just now. Okay, so the steps. Write down the formula of the positive and negative ions and then count the positive charge and negative charge, what we do. If not equal, we will add more until they are equal. Like polyatomic ions, we will put in bracket. Yeah, like this one. Polyatomic, we will put in bracket. Okay, let's go through some example. Yeah, sodium. Okay, yes. Okay, so we know sodium is plus 1. Okay, carbonate is 2 negative. So we need sodium, 2 of them. Carbonate is 1 to get the balance charge. So the correct formula, Na2CO3. Okay, magnesium hydroxide, magnesium 2 plus, hydroxide negative 1. So we need 2 of them. So the correct formula, magnesium hydroxide, 2 of them. Okay. Okay, another one. Okay, aluminum oxide. Yeah, we did this just now. 2 aluminum, 3 oxygen to balance the charge. Okay, Al2O3. And then iron 3 sulfate. Yeah, iron 3. Iron 3 sulfate is 2 negative. So which means we need 2 iron sulfate. We need 3. Okay, so that will give us plus 6, negative 6. So we will have Fe2SO4 3 unit. Okay, like that. Okay. Yes. Yep. Okay. Okay. Structure of ionic substance. Okay. So just now we already know. Example, sodium plus and chlorine negative, they will attract each other because they have positive and negative charge. They will attract each other. What if we have millions of them, millions and millions of them? They will arrange like this. Yeah. Okay. The positive and negative, they will arrange side by side, millions and millions of them. They will form this giant ionic lattice. Okay, it's a regular arrangement of particles in a repeated pattern. Okay, so in this case, we have the sodium chloride lattice. Okay, whereby in between they have strong electrostatic attraction. Yeah, we have this oppositely charged ions with a strong electrostatic forces of attraction. Yeah, okay. Yes. Okay. So if we have this kind of attraction, they are very, very strong attraction. So what kind of properties we expect from these ionic substances? Okay. We expect these ionic substances, they will have high melting points, high boiling points. Okay. Because of this strong attraction. Very, very strong. And then we have millions and millions of them. Okay. So they will be having high melting and boiling points, good electrical conductivity when aqueous or molten. Okay. Yeah. So when they are in solid, this charge, they cannot move. Okay. So they cannot move. So they cannot conduct electricity. But if we dissolve in water, so they are free to move. Yeah. So they can conduct electricity, or we melt it. Okay. If we melt it again, they are free to move. They can conduct electricity. So only um, when they are aqueous or molten, they can conduct uh, uh, electricity. But when in solid, it's poor electrical conductivity. Okay. Yeah, we see this yellow box again. Means for core, we just need to know this. Okay. But for extended, we need to know the explanation as well. For core, we just need to know the properties, these three properties. For extended, we need to know the reason as well. Okay, we know that. No problem. We know all of the reasons. Okay, so we can take a look. Okay. Yeah. Okay, all of the reasons. Okay, the reason high melting and boiling points is because of the strong electrostatic forces of attraction. When we have positive and negative charge together, they will attract. So this is what we call electrostatic forces of attraction. Okay. They can conduct electricity because they have moving charge particles. Yeah. Okay. When these particles, we break the bonds, they are free to move. They can conduct electricity. So solid state, they cannot move. They are fixed. Okay. So they are poor conductor in the solid state. When they are dissolved in water or they are melted in the molten form, they can move. Okay, so they can conduct electricity. Okay. Yes. Yes. We will look at oxidation numbers. Okay. So sometimes we need to determine whether we have a oxidation reaction or we have reduction reaction. Okay. So last time we already know if we increase oxygen, it is oxidation. Okay. If we decrease oxygen, it is reduction. Okay, so we can see here, this one, it is increase oxygen. So we know this is oxidation. Okay. Yeah, but we have another way. We look at the oxidation number. Yeah, okay. If we look at the oxidation number, we can also determine whether it's oxidation or reduction. If oxidation number increase, it is oxidation. If oxidation number decrease, it is reduction. Yeah, okay. So we can see here, this ion is +2, the oxidation. Now it become +3. We see this Roman numeral. This Roman numeral is telling us the oxidation number. Yes, okay. So going from +2 to +3, it is oxidation. Yes, okay. So this is how we um, recognize that this is an oxidation reaction. Okay. So now um, the oxygen here, it is oxidizing agent. Why? Because it oxidized this ion from +2 to +3. So this oxygen in this case, it is a oxidizing agent. Okay. And then, yes, how we work out the oxidation number. Okay. So there are some rules to work out the oxidation number. So just now, we look at this Roman numeral is one way. Okay. Next, we will follow this rule. Okay, uncombined element is always 0. Okay, so let's say iron 0, the atom before chemical reaction, this is 0. Okay, and then nitrogen, so all of the elements, okay, before any chemical reaction is 0. Okay, uh, fluorine 0. Okay, neon 0. Okay, before any chemical reaction is always 0. Okay, and then for some compounds, okay, so once we have chemical reaction, we have oxidation number. Group 1 is always plus 1. Okay, so we see here, group 1 is always plus 1. Group 2 is always plus 2 after it formed the ion. Okay, and then group 6, oxygen is always negative 2. And then group 7 is always negative 1. Okay, yeah, hydrogen is always plus 1. Group 2, always plus 2. Aluminium, always plus 3. Fluorine, always negative 1. Group 7, yeah, just now group 7. Yes, and group 6, oxygen always negative 2. So we follow this rule. Okay, monoatomic ion is always equal to the charge. Okay, copper plus 1, the oxidation number also plus 1. Sulfur 2 negative, oxidation number negative 2. Okay, so now if we have polyatomic ion, will be equal to the charge. Okay, so example, so this one, SO4, the total charge is negative 2. Okay, if we have compound, let's say sodium chloride, plus 1, negative 1, total is 0. Okay, MgF2 plus 2, negative 2, always 0. The oxidation number of a compound, the total is always 0. Okay, overall is always 0. Okay, so now we will look at some example. Yeah, N2, N2O4, NO3 negative. Okay, now this N2, it is element. Okay, so it is element, so means the oxidation number is always 0. Okay, N2, 0. Okay, so let's say it undergoes some chemical reaction, it may become 3 negative, for example. Okay, but at the element situation is always 0. Okay, N2O4, this is a compound. Yeah, N2O4 is a compound. And then oxidation number of compound is always 0. Okay, so the total is always 0. Total is always 0. And then oxygen is always negative 2. Okay, so the total is always 0. And then oxygen is always negative 2. So we can see here, oxygen is always negative 2. So we have 4 oxygen, 4 negative 2. 4 times negative 2. And then nitrogen, we have 2 of them. So which means we have 2 times nitrogen plus 4 times oxygen. Okay, is equal to 0 because overall of a compound is always 0. Okay, so 2 times nitrogen. So we want to work out the oxidation number of nitrogen. Okay, and then oxygen is negative 2. Negative 2 equals to 0. So 2 times n, okay, 2n, 4 times negative 2 minus 8 equals to 0. So means 2n equals to plus 8, algebra, okay? So n equals to 8 divided by 2 equals to plus 4, okay? So in this case, we can work out the oxidation number of nitrogen is plus 4, okay? Based on this calculation. Okay, next. Okay, you see, just now the N2O4 is 0. Okay, but this one, we can see negative charge here. So which means this NO3 overall is negative 1. Okay, So we have 1 nitrogen plus 3 oxygen is equal to -1 because overall is -1. So this nitrogen plus 3 oxygen is -2 equals to -1. Okay, and then n 3 multiply -2 minus 6 equals to -1. N equals to 6 minus 1 equals to 5. So in this case, the oxidation state is of nitrogen is +5. Okay, so which is telling us this nitrogen, it can have different oxidation state, depend on the situation. It can be 0, it can be +4, it can be +5, depend on the situation. Okay, so we can calculate it based on the rules that we get. Okay, and then next, okay, so final point for today, okay, we'll be looking at definition of redox reaction. Okay, so just now we already know if we increase oxygen is oxidation, okay, decrease oxygen is reduction. Okay, how about hydrogen? Okay, hydrogen is actually negative. Okay, so hydrogen is negative. If we increase hydrogen is reduction. If we decrease hydrogen is oxidation. Okay. But how about if we do not have oxygen or hydrogen in the chemical reaction? Then we cannot use this. We must use oxidation number. Okay. So that's why just now we study about the oxidation number. Yeah. Okay. So we can see here this iron is going from +2 to +3. Increase oxidation number. It is oxidation. Okay, how about chlorine? Chlorine is going from 0 to -1. It's a decrease of oxidation number. So this is a reduction. Okay, oxidation and reduction is opposite. Okay, and then another thing we can see here, the iron, it lost electrons. Losing electron is oxidation. Okay, in this case, chlorine is the oxidizing agent. It oxidizes the iron from +2 to +3. Okay. Yes. So, summary, oxidation is electron loss. Oxidizing agent will be electron acceptor. Okay. They are accepting electrons like this chlorine, accepting electrons. Okay. Oxidizing agent. Okay. And then, yeah, reduction is electron gain. Reducing agent will be electron donors. Okay. So we have a transferring. Okay. Electron transferred from reducing agent to oxidizing agent. Okay. Reducing agent will be oxidized and oxidizing agent will be reduced. Okay. So we can see this summary here. Oxidation, reduction. If this happen, gain of oxygen, loss of hydrogen, loss of electron, increase oxidation number, it must be oxidation reaction. Loss of oxygen, gain of hydrogen, gain of electron, decrease of oxidation number, it will be reduction. Okay. And then finally, final point for today, we can test for oxidizing and reducing agent. Okay. So we can use potassium manganate to test reducing agent. Okay. So if we have reducing agent, the color will change from purple to colorless if we have reducing agent. Okay. How about if we want to test for oxidizing agent? If we want to test for oxidizing agent, we will use this potassium iodide. Okay. It will go from colorless to brown. Okay. If we have this oxidizing agent, colorless to brown. Okay. Yes. Okay. So the time is perfect. We can stop here. So tomorrow we will continue with the what it means by covalent bonding. Yeah. Okay. So this is our progress. So unit 3 in progress. Okay. So yeah. So if no other question, we will stop here. Yeah. Okay. We continue tomorrow. Okay, thank you. Bye bye. Bye bye.